In this video, I'm going to walk you through the plate count technique, and by necessity, I'm going to review serial dilution. So why do we do serial dilution and why do we do the plate count technique? First, the plate count technique is a way of quantifying the number of live microorganisms in a sample. And so it's like just asking how many live cells are in this liquid. So we do this for a couple of reasons. Sometimes we want to know in the environment how contaminated is something, or how many cells are growing in something. Sometimes in a laboratory, or very often in a laboratory, we need to standardize numbers of cells between experimental treatments. And so we always need to know how many cells are in a sample in a laboratory. So we do this a lot. So this is a technique, again, for quantifying the number of living cells. And the way it works is we put some amount of our unknown culture that has the unknown number of cells we want. We put some of it onto a petri dish. So if I draw a little petri dish, and wherever the little cells end up, if we incubate this, they will form colonies, and we will be able to count the colonies, and then we will know, we can do some math to figure out how many were in the original sample. So I'm going to show you sort of what that looks like. So let's see if, yeah, okay, cool. So what we're going to do is go through an example of what it looks like when we do the plate count. And I'm going to go through the whole serial dilution, and we will use sort of made up numbers to see what it looks like. Now, on that petri dish that I drew, there were something like 10 colonies. And if there are a small number of colonies, like 10 colonies, we worry that small variations will look too big. So 10 versus randomly 12 will look meaningful when it might not actually be. Don't worry about it. It's just that statisticians want to have big enough numbers that we don't worry about statistical noise. So in order to count colonies on a plate and know that the number we get is meaningful, we need at least 30 colonies. And at the other end, if we put too many cells on a plate, they form basically one giant colony. And so we need under 300 colonies. So if you, it's entirely possible to have 600 colonies on a plate and count them. The problem with this is there's a high probability that a lot of those colonies were founded by two cells on top of each other. And so you start to get an inaccurate number if you have more than 300 colonies or less than 30. Okay, so this is our target. And the tricky thing is that usually a bacterial culture, we just don't know how many are in there. And so we don't know how much volume of this to put on our petri dish to give us between 30 and 300 colonies. So we do a serial dilution. So I'm going to first make a bunch of dilution tubes, and we will use these dilution tubes. And I'm also going to make a bunch of petri dishes. So we'll see the petri dishes. So I've drawn 5 dilution tubes. Now each of these is full of some kind of sterile liquid that I can use to dilute my original sample. And let's call the original sample our unknown. And instead of calling it our unknown culture, let's just call it our, let's just make this a little more elaborate and correct and call this our sample with unknown population size. So population size is the number of cells. So our sample with unknown population size, that's this. We're trying to figure out the population size using the plate count technique. So we are going to, well, we're also going to put a cap on this to keep dust out, right? Of course, we always cap this. So we are going to use these 5 dilution tubes in series to make different dilutions of our sample with unknown population size. And so each one of these tubes has 9 milliliters of sterile broth. So the way this works is from this to this first one, I transfer 1 milliliter. Now, I could do this with any other volume. I could choose other volumes if I wanted to, but this is the simplest way. 1 milliliter into 9 milliliters. And so what that's going to give me is a final volume of 1 plus 9 equals 10 milliliters. Okay, so this is going to have a total of 10 milliliters in it once we add our sample. And we will have diluted our sample with unknown population size tenfold. So because we have taken 1 milliliters of it and redistributed it into a total of 10 milliliters. So let's start using numbers. So we're going to say, for the sake of this exercise, let's say that this sample with unknown population size actually has 6 times 10 to the 7th cells per milliliter. Now, the person doing this serial dilution doesn't know this number. This is what they want, but we know it because we're making it up. So 6 times 10 to the 7th cells per milliliter. Well, if you take 1 milliliter and put it in here, how many cells is that? Well, that is a total of 6 times 10 to the 7th cells. Let's see, times, or cells per milliliter. That's our units. Times 1 milliliter, because that's how many cells we put in, or how much volume we put in. We can cancel milliliters, and that gives us 6 times 10 to the 7th cells. Just cells, right? So if we multiply a volume times a concentration, we get a mass, or in this case, cells per mil times mils gives us cells. So there are 6 times 10 to the 7th cells in here. That's how many we put in here. But the volume in here is no longer 1 milliliter. So now this many cells is in 10 milliliters. Okay? So we have put this many cells and we've resuspended them basically and dispersed them into 10 milliliters. And so this gives us a concentration of 6 times 10 to the 6th cells per milliliter. That's the concentration in this tube. Now, compare this number, which is the concentration in this tube, to this number, which was the concentration in our unknown sample. So this was 6 times 10 to the 7th cells per milliliter. And this is now 6 times 10 to the 6th cells per milliliter. And so if you've gone from 10 to the 7th to 10 to the 6th, that's a tenfold dilution. So we have diluted this tenfold. So now I'm going to erase a bunch of stuff. I don't know if this will work. Let's try it this way. So I'm going to relabel some things. Yeah, there we go. So I'm going to label this the 1/10 tube, or the 10 to the minus 1 tube. Those mean the same thing. 10 to the minus 1 is 1 over 10. So this is the 1/10 tube because the concentration went down by a factor of 10. So this is the dilution, and then this is going to be the concentration in this tube. 6 times 10 to the 6th cells per milliliter in the 1/10 tube. The next step is to take 1 milliliter of this and put it into the next tube. And the same thing is going to happen. 1 milliliter of this plus 9 milliliters of sterile broth equals 10 milliliters. So we've taken however many cells are in 1 milliliter of this and diluted them into 10 milliliters. And so again, that's a 10-fold dilution. So we call this the 1 to 1/100th, or 10 to the minus 2 tube. And it's going to have 6 times 10 to the 5th cells per milliliter. It's just 1/10 of this one. And this is 1/10 of our sample with unknown population size. And so we do the same thing. 1 mil into here, dilute it 10-fold. Do the same thing with this one, and do the same thing with this one. And that's a series of dilutions, and each time we get a 10-fold lower concentration. So the concentration in our 1 to 1,000, or 10 to the minus 3, is going to be 6 times 10 to the 4th cells per milliliter. In our 1 to 10,000, or 10 to the minus 4, is 6 times 10 to the 3rd cells per milliliter. And last, this would be a 1/100,000th dilution, or 10 to the minus 5th, and that's 6 times 10 to the 2 cells per milliliter. So we have just figured out the concentration in all of these tubes. And the concentration of cells in this tube is 6 times 10 to the 2 cells per milliliter. That's what that says. And anywhere in between, we can come up with what that is. This is a hypothetical example, so the math is perfect and the concentrations are perfect and there's no statistical noise or anything like that. So our goal, again, was to put these on a petri dish and count the number of colonies we got. So we put 100 microliters, or 1/10 of a milliliter, onto a petri dish. So how many colonies will we get on a petri dish? Well, let's do the example with this one first. So if we take 6 times 10 to the 6th cells per milliliter, and we put 0.1 milliliters onto a petri dish, we can cancel milliliters, and that's going to give us 6 times 10 to the 5th cells, and that would be 6 times 10 to the 5th colonies on a petri dish. So ideally, each cell makes one colony. And 6 times 10 to the 5th is 600,000. Remember, we are trying to get a number of colonies that's between 30 and 300. So, have we achieved that? Is this between 30 and 300 colonies? No, this is 600,000 colonies. So what we're going to get here, that's what we would get if we were to put 1/10 of a milliliter of this dilution onto a petri dish, right? That's what we calculated here. If we were to use this one instead, we would get 1/10 this many. And if we use this one, we would get 1/100th. This would get 1/1000th of these. So let's look at what we would get. Let's just figure out how many colonies we would get from all of these. So this one would give us 6 times 10 to the 5th colonies. This would be 6 times 10 to the 4th colonies, 6 times 10 to the 3rd colonies, 6 times 10 to the 2 colonies, 6 times 10 to the 1 colonies. Okay, so which one of these is between 30 and 300? It might be none of them. But let's see. This is 600,000, so that's too high. 60,000, too high. 6,000, too high. 600, too high, 60. That's a number we will accept. So if we were to put, again, 1/10 of a milliliter of this onto a petri dish, we'd get a good number of colonies we could count. And I should say that 1/10 of a milliliter number, what we put on a petri dish, that is just a good amount of water that will not change the characteristics of the agar. The water in that sample will be fully absorbed, and so that's a standard amount that we'd put on a petri dish. So by doing this serial dilution, we achieve a concentration within a range we can count. And so again, this is a hypothetical example. And let's say that you and I know this number, and you and I have done all this math to figure out all the concentrations in all these dilution tubes. And you and I know how many colonies each of these dilution tubes are going to make. So we go ahead and we make all the petri dishes. And we can't count this. It's 600,000, and this one's 60,000, and this is 6,000, and this is just like too many, and this is like 60. So we get our friend and coworker to come in and count them, and they say, okay, too numerous to count. That abbreviation, too numerous to count. Again, too numerous to count, too numerous to count, too numerous to count, 60. And so we asked them, okay, well, how many cells were in the original sample with unknown population size? What was the population size? Based on this number, well, they would have to know the dilution. And they would, because we would have labeled these plates 10 to the minus 1, 10 to the minus 2, 10 to the minus 3, 10 to the minus 4, 10 to the minus 5. So our colleague has gone through and counted the colonies on these plates and needs to figure out what the unknown population size is. Okay, so the way this calculation works is you would take whichever number you get, so you can ignore all this. That's the number of colonies. So we say 60 colonies, and divide this by the volume that went onto this. So first we're just thinking how much volume from this dilution, and that volume is always 0.1 milliliters. So 60 colonies divided by 0.1 milliliters. And then we also incorporate this dilution factor. And so the dilution factor and the volume you added and the number of colonies is all you really need. And the way we deal with the dilution factor is we divide by it. So times 1 over 10 to the minus 5th, that's the dilution factor. And there are different people will do this in a different way, but they're all essentially doing the same mathematical thing. And so this is the same as saying 60 colonies divided by these, multiply them by each other, 10 to the minus 6, which is the same as saying 60 times 10 to the 6th. 10 to the minus 6 is 1 over 10 to the 6th. And we want to put this in scientific notation, so we move the decimal point, which would be here after 60.0 or whatever, move it over 1. So it'd be 6.0 times 10 to the, and we move that over 1 to the left, so add 1 to that. 6.0 times 10 to the 7th. So our colleague could tell us, well, I think the unknown sample, or the sample's unknown population size is actually 6 times 10 to the 7th. And the units there Well, in this calculation, what they would come up with is colonies per milliliter. Now that's a strange unit, right? Because what we really wanted to know was cells per milliliter. But we don't know cells per milliliter, we just know how many colonies were formed on this plate. So there could have been a bunch of dead cells in there that we just don't detect this way. And so actually what we say, because this is silly, there aren't any colonies, what we say instead is 6.0 times 10 to the 7th colony forming units per milliliter. That's the actual unit you would see in a publication. That's what we use. And so we shorten this to 6.0 times 10 to the 7th CFU per milliliter, colony forming unit per milliliter. Okay, so again, this number we could calculate from the number of colonies, the dilution factor, and the volume that we placed on the plate. And that volume is usually a standard volume. It's almost always 0.1 milliliters. And then the dilution factor is, it's incorporated into these, and you label the plates with the dilution factors that they correspond to. So someone who didn't set this up, but just counted colonies and could see these dilution factors, they could do this calculation. That's pretty much how it works. That's the plate count technique, and going through the whole process of the plate count technique and doing the calculation. So we'll come back in the next video and maybe do some practice calculations so you can see how that works, and then we will do something different.